Gel electrophoresis. Get the two cords out of the cabinet. These cords may or may not be connected. One should say switching mode power supply in all capital letters under a red and white Meanwell logo. The other should look like a regular cable. One end must fit into the jack of the first cord, while the other end should be a typical three-pronged wall plug. Get the Invitrogen gel rig out of the cabinet. These are the first two parts, the UV viewing shield and the eGel Safe Imager. This is the eGel iBase. If the parts are not already attached, then the Safe Imager should be placed on the table. The iBase should be placed on top of that so that the cylinder legs of the iBase fit into the indentations on the Safe Imager. Connect the two cords. Insert the cylinder-ended jack of the switching mode power supply into the DC in opening on the back of the Safe Imager. Insert the wall plug into an outlet. Also, check that the wire coming out of the Safe Imager is connected to the back of the iBase. Obtain an EX Agarose 2% gel. It looks like this, and the gels are located in a box next to the micropipetters, which are next to the window looking out into the hallway. Remove the gel from its packaging by tearing it open at the top, beginning with a small indent on the left side toward the top. Place the gel in the rig by sliding the gel in from the left side until it clicks underneath the gray object sticking up. Carefully pull out the comb. It is made of clear plastic and it covers the rectangularly shaped holes, these are called wells, located at the top of the gel. This is one of the samples that is going to be run in the gel. Obtain a micropipetter and the appropriate tips. Pipette whatever amount of your sample the procedure calls for into the first well. For help on how to pipette solutions, talk to Ms. Gleicher, Ms. Villa, Ms. Arcus, or another student. It works just like an eyedropper, it's just fancier and more precise. Do the same for the rest of the lanes if you have additional samples, loading the same amount of sample into each of the lanes. You may also have to use loading die. Consult your procedure or your mentor for more information about this. Before loading water into the extra lanes, you want to take one lane and load a DNA ladder into it. This allows you to make sure that the bands that you are seeing are the appropriate amounts of nucleotides. Now, instead of leaving any lanes empty, pipette distilled water into those lanes. Pipette the same amount of water as you did for the samples. Take the orange UV viewing shield and place it on top of the iBase so that it clicks into place. Press the red Go button. Press the other red button at the bottom of the Safe Imager at any point when the gel is running to see the bands. If you want to take pictures of the bands, you must do that when the UV light is on and the UV viewing shield is also on. Press the red button again to turn it off. When the gel has finished running, it's probably best to take pictures. Remove the orange viewing shield after this is done, and carefully take the gel out of the iBase. At this point, you have two options. 1. You may store the gel in the refrigerator, or 2. You may throw it away in a garbage can. Disconnect all the wires. Place them in the cabinet located above and to the left of the micropipetters. Place the gel rig and all three of its parts next to those cords. Polymerase chain reaction, otherwise known as PCR. Locate the machine. It is black and blue with a rectangular screen on the front. It is located to the right of the centrifuge and to the left of the large window looking out from the STEM lab into the hallway. Turn it on. The switch is located on the back of the machine in the upper right-hand corner. Wait for the machine to finish its self-diagnostic program. The self-diagnostic screen will say communications, thermal engine, block, etc. When that disappears, proceed to the next step. Note, if it says fails, as it did just now, turn the machine off, wait a few seconds, and then turn it on again. The screen is a touchscreen. Press Program at the top of the screen. Press Write New Program. Press Standard. Press Open. Adjust the temperatures according to whatever your mentor or protocol says. To change something, temperature, time, etc., simply press it, enter the new number, and then press Store Temp or Time. When all of the settings are satisfactory, press Save As in the bottom right-hand corner of the screen. Find a program that says empty in all capital letters next to it. If such a program is not visible, press the large arrow pointing downward. Press the word empty. Press Save As. Type in the name that you want to call your program. Press Enter. To run that program, press Run at the top of the screen. Press Load Another Program. Use the up and down arrows, if necessary, to find your program. Press it. Press Open. Open up the lid by pressing the blue button located above the screen and lifting the lid upward. Load your samples into the wells. Be very careful that the lids of your PCR tubes are completely closed, or else the samples have the potential to evaporate. Close the lid by pushing it down until you hear a click. Press Start Current Program. When the thermal cycler is finished running, approximately 3 hours later, press End Program, wait a few seconds, and open the lid. It is okay to take the samples out after more than 3 hours. Take your samples out and store them in a freezer, or run a gel, purify the samples, etc. Turn off the machine by flipping the same switch that you used to turn it on.